we are going to start next experiment in pharmacology and toxicology, in which experiment number 20, to observe and study the effect of convulsant and anticonvulsant in mice by using CD of MSBT. The biological name of mice is mus musculus, in which epilepsy, convulsion, anticonvulsant. This part where we already seen in theory. Convulsion means there is a sudden uncontrolled electrical disturbance in the brain, or it is related to CNS disorder. So to treat this disorder, the agents are used. They are named as anticonvulsant agent. And convulsion means these are the agents they produces convulsion. Anticonvulsant, they act against the convulsant agent, means they both are antagonists of the each other, means one drug stops the action of another drug. So in which preposition first? If epilepsy, it is a group of disorder of CNS. It is characterized by paroxysmal cerebral dysarrhythmia, manifesting as a brief seizures. Seizures means it is a sudden uncontrolled electrical disturbance occur in the brain, and it is occurred due to there is interruption or there is misconnection in between the nerve cell and the brain. So there is connection in between the brain and nerve cells, which transport impulses, where main work of these impulses. So in which in seizures condition there is a sudden and uncontrolled electrical disturbance occur in the brain. Paroxysmal sudden. Cerebral dysarrhythmia and manifesting a brief episodes, that is seizure and loss or disturbance of the consciousness is occur without characteristic of body movement. Means when epilepsy occur, there is sometime maybe chances to different types of the body movement, and sometime in other cases there may be no body movement. So this is about epilepsy. So epilepsy, convulsions, they are CNS disorders. So next point is types of the epilepsy, in which generalized seizures, tonic-clonic seizures, or grand mal or major epilepsy. Major epilepsy, it is also known as tonic-clonic seizures or grand mal epilepsy. Then second, absence seizures or petit mal or minor epilepsy, atonic seizures, myoclonic seizures, and hypersarrhythmia, that is infantile spasms. Then parietal seizures, in which simple parietal and complex parietal seizures. So these are the types of epilepsy. So to treat this disorder, a group of CNS disorder, it is treated by using a pharmacological agent is known as anti-epileptic agent. So drugs used in the treatment of convulsion are known as anti-epileptic drugs. The next phase of convulsion, in which tonic, clonic, Straubus, and flexon. These are the phases of convulsion. So by this way, or by these phases, convulsion is occurred. Then preposition for Straubus tail. Erection of tail in S-shaped structure is known as Straubus tail. So mice, when some convulsant agents are administered to this animal, then the symptoms may be occurred. That is, which shaped tail? Structure of this tail is S-shaped. So erection of the tail in the S-shaped structure is known as Straubus tail. This symptom is observed after the administration of either convulsant or anticonvulsant drug. The next point is mechanism of action of diazepam. So diazepam, it mainly acts as an agonist. Agonist means ability to bind with the receptor. So it having ability to bind with the GABA receptor and potentiate the action of GABA. Potentiate means to stimulate the action of GABA. GABA is gamma aminobutyric acid receptor. So this diazepam act as an agonist and bind to the GABA receptor and potentiate the action of this receptor. So GABA generally they are having the inhibitory in nature. They are having the inhibitory action on the central nervous system. So this is about mechanism of action of diazepam. The next point is procedure for the experiment. So we discuss this procedure from CD of MSBT. Effect of convulsant and anticonvulsant in mice or rat.
The objectives of this experiment are to understand the terms epilepsy and convulsions or seizures, to know classification of seizures and its behavioral manifestations, to know that electroencephalogram, that is EEG, is employed to detect neuronal excitability, to understand that epileptic seizures can be evoked in normal brain by kindling, that is, by electric shock or chemical convulsant, to know that anticonvulsants are employed in the treatment of epilepsy.
We all know that what is meant by epilepsy, convulsions, seizures. So these are the objectives of this experiment. And requirements: animal albino mice, requirements in which syringe and needle, cotton, weighing balance, drug, strychnine sulfate 4 mg, IP solution, diazepam 2 to 4 mg. The next theory behind the experiment.
Epilepsy is a group of CNS disorder characterized by brief episodes of seizures. Seizures are also referred as convulsions. Convulsions are paroxysmal episodes of spontaneous, uncontrolled neuronal discharge and are characterized by tonic body movements and or disturbance in consciousness along with change in motor, sensory or behavioral activity.
Epilepsy is a group of central nervous system disorder when there is disturbance into the CNS, either in their motor activity, sensor activity, or in the behavioral activity. This change occurs due to there is interrupt or misconnection in between the impulses transmission through the neurotransmitter, or there is interruption in between the connection of nerve cells and the brain. Then this disorder is occurred. Seizures are also known as convulsion and also known as epilepsy. Convulsions are paroxysmal. Paroxysmal means they are either sudden type, suddenly they are increased, or it is an attack, or it is an involuntary movement. Also they are uncontrolled neuronal discharge, means there is neuronal discharge from the CNS and it is uncontrolled discharge during this paroxysmal or during this seizure disorder. So it is characterized by tonic body movement and or disturbance in the consciousness. When seizure is occur or epilepsy is occur or any individual suffers the epilepsy disorder, then there is disturbance in the consciousness of this individual, and along with some change are observed in their motor activity, sensor activity, or there may be change in the behavioral activity. As shown in this figure, when seizures or epilepsies occur to any individual, then their condition like this. Is it visible in your screen?
Yes, ma'am.
Ma'am.
So when CNS disorder is occur, then there is change in the motor, sensory, as well as in the behavioral activity.
The electric shock induced convulsion in animals represent grand mal epilepsy. Chemical induced convulsions with strychnine represents petit mal epilepsy. Chemical induced convulsions are characterized by tremors, hyperexcitability, clonic phase followed by tonic phase, and deep abdominal respiration is seen. This episode of convulsions results from either decrease in concentration of GABA, glycine, or an increase in concentration of acetylcholine, glutamic acid. Strychnine, a CNS stimulant, inhibits glycine and induces convulsion in animals. Anticonvulsants like diazepam acts as an agonist of GABA and inhibits spread of abnormal transmission in brain.
The electric shock is induced, means produced a convulsion in the animal represent grand mal epilepsy. if taken an animal, either it is mice or the rabbit. Albino mice is used in this experiment. So if produces electric shock, through the electric shock, convulsions are produced in the animal. Then epilepsy is produced by this animal or shown by this animal is known as grand mal epilepsy. Means when shock is given to that animal, then it is susceptible to or produced to a convulsion or epilepsy. So such type of epilepsy is known as grand mal epilepsy. The next one, chemical induced convulsion, means convulsion produced in the animal with the help of chemicals, means chemicals injected to this animal and convulsions are produced in this animal. So such type of the epilepsy is known as peptimal epilepsy. So strychnine is used to produce an convulsion. Chemical induced convulsion, that is chemically produced convulsion or epilepsy, it is characterized by tremors, hyperexcitability, clonic phase, which is followed by tonic phase, and deep abdominal respiration is seen. Means there is disturbances in respiration is occurred. Hyperexcitability means hyperactivity, that is behavioral change, and tremors again they are related to seizures, that is involuntary movements. So these phases or characters or symptoms observed when convulsion is produced by using an chemical. Then this episode of, that is the seizures of the convulsion result either decrease in concentration of GABA. So because of this convulsion, there is decrease in the concentration of gamma aminobutyric acid, also glycine, and increase the concentration of acetylcholine and glutamic acid. When convulsions are produced in any individual, then there may be two changes is to be occur. Firstly, there is increase in concentration and decrease in concentration. So decrease in the concentration of GABA and glycine, while increase in the concentration of acetylcholine and glutamic acid. Then strychnine as a CNS stimulant, it inhibits the glycine and produces convulsion. So strychnine, it is an agent, a CNS stimulant, which inhibits. CNS stimulant means they stimulate the central nervous system and they inhibit the glycine and produces convulsion. So activity of this glycine is inhibited by this drug and they produces convulsion in the animal. Then anticonvulsant agent anticonvulsant agent like the diazepam act as a agonist of GABA. So this diazepam act as agonist of the GABA and inhibit the spread of this abnormal transmission in the brain. When it stops the spread of abnormal transmission in the brain, it helps to recover this convulsion disorder. So this is about theory behind this experiment. So from the theory, we concluded that anticonvulsant agent, convulsion produces convulsion, and anticonvulsant agents are used to treat this convulsion or to stop the convulsion symptoms. The next point is procedure.
To study the effects of convulsant and anticonvulsant in mice, select 2 adult healthy mice weighing not less than 20 gram and label them as mice A and mice B. Observe their normal posture and normal muscle tone, particularly in limbs and tail. Now inject 0.1 ml of stock solution of strychnine hydrochloride to mice A intraperitoneally. You can see that strychnine induces clonic convulsions characterized by Stroop's tail, that is S-type erection of tail and jerky movements of whole body. This clonic convulsions finally leads to death of the mice A. Inject 0.2 ml of diazepam solution intraperitoneally to the mice B. After half an hour, administer 0.1 ml of strychnine sulfate solution intraperitoneally to the mice B and observe the onset and severity of convulsions. You can see that severity of convulsions such as Stroop's tell whole body movement is less in this mice. This suggests that diazepam protected the mice from the convulsions induced by strychnine. Clinically, diazepam is useful in different forms of epileptic seizures and also as a drug of choice in status epilepticus.
So by a stepwise procedure, first we select any healthy mice, either any sex, then they labeled, after that inject and strychnine sulfate. So we concluded that from this procedure, when by selecting an intraperitoneal route and inject strychnine sulfate to this animal or the mice, it shows Stauber's tail jerky movements. Jerky movements means very sudden or quick movement is to be observed. That is jerky convulsion. Then there is severity of the conclusion is to be occur. And after the administration of diazepam and strychnine sulfate, then it produces there is no Stauber's tail, no jerky movement is to be observed. As such, animal observed in the normal movement.
And normal muscle tone, particularly in limbs and tail. Now inject 0.1 ml of stock solution of strychnine hydrochloride to mice A intraperitoneally. You can see that strychnine induces clonic convulsions characterized by Stroop's tell, that is, S-type erection of tail and jerky movements of whole body. These clonic convulsions
Okay, so this condition is known as jerky convulsion. So strychnine sulfate or strychnine hydrochloride produces jerky convulsion as well as Stauber's tail and severity of the convulsion. So it produces jerky movement, that is strychnine sulfate. It produces convulsion. So it is known as convulsant agent. And after which route is selected for injection or administration of strychnine sulfate?
Intraperitoneal.
Okay, so this movement of this animal, it is jerky convulsion.
of the mice A. Inject 0.2 ml of diazepam solution intraperitoneally to the mice B. After half an hour, administer 0.1 ml of strychnine sulfate solution intraperitoneally to the mice B and observe the onset and severity of convulsions. You can see that severity of convulsions such as Stroop's tell whole body movement is less in this mice. This suggests that diazepam
Okay, so after the administration of diazepam, there is Stauber's tail jerky movement observed or not?
No.
protected the mice from the convulsions induced by strychnine. Clinically, diazepam is useful in different forms of epileptic seizures and also as a drug of choice in status epilepticus.
So diazepam is a drug of choice in status epilepsy. It is one of the type of epilepsy, status epilepticus. Diazepam, it acts as an anticonvulsant agent and do not produces convulsion. And if there is convulsion produced, then it helps to inhibit or to treat the convulsion. That's why here we concluded that from the procedure, therefore, strychnine sulfate sulfate, it produces convulsion. That's why it is convulsant agent, and diazepam, it acts as an anticonvulsant agent. The next point, observation. So you have to write same observation table in your manual as well as in your report. Strychnine sulfate. Then onset of time 50. It is in the second. Then nature of convulsion. There is Strauber's tail, jerky, movement, clonic convulsion. Death or recovery, death after the 3 minutes. Jerky movements and Strauber's tail occur होती है. It means that death will be occur. Then there is no Strauber's tail. मग इथे death होईल का diazepam strychnine after the administration of diazepam?
No.
No, because diazepam acts as an anticonvulsant agent. It relieves the animal from the convulsion. So there is no Strauber's tail, minor body movements, minor clonic convulsion, no death of animal. It is protected from the convulsion. Duration of convulsion is less. Reduction in time of extensor phase of convulsion. So you have to write this observation table as such.
You have learned that convulsions induced by chemical agents produce clonic type contractions characterized by jerking of head and body and Straub's tail. Anticonvulsant activity of a drug under test is complete protection from convulsions.
So we concluded that anticonvulsant agent act against convulsion, that is to treat the convulsion. And convulsion, they are produced by using either they are produced by electrical shock or they are produced by administration of some chemical agent like strychnine sulfate. And after their administration, during the convulsion, they showed symptoms or characterized contraction like jerky head and Strauber's tail. So this is about today's experiment to study the effect of convulsant and anticonvulsant on the mice. Okay, so here you have to write onset time. 50 seconds, 80 minutes. Then characters, Strauber's tail, jerky movement. There is no jerky movement, no Strauber's tail. Then recovery, about recovery, death after the 3 minutes, and here no death of animal. Animal is protected from the convulsion and there is less convulsion, reduction of time of extensor phase of convulsion. And lastly, conclusion, strychnine sulfate, it produces convulsant and diazepam, it produces anticonvulsant effect. And after that, attend these questions. So here we complete experiment number 20.